Hello, Miss Francis and future me, or anybody who's watching this video. Today we're going to be talking about the plasma membrane and transport in the neuron and kidney. Now the question we're addressing is how do organisms use different kinds of transport to maintain homeostasis? Now the first way is through neurons and the action potential, and the second way is through osmoregulation and kidney function. The picture right here is a picture of the neuron, and we're just going to label all of the parts of the neuron. So the first part is the dendrite. We have the nucleus, which is inside of the cell body. Here we have the myelin sheath, which is located on the axon. The axon refers to this part of the neuron. We have the Schwann cell, which is located in the myelin sheath on the axon. And then we have the node of Ranvier. And then we have the axon terminals. So now we're going to talk about the different functions of the different parts of the neuron. So the first part is the axon, and it is the conducting fiber. And I like to say it's where all the action happens because that's where the action potential travels through the neuron for cell communication. Then we have the Schwann cell, which produces the myelin that forms the myelin sheath, which creates insulation around the Schwann cell and allows action potential to move quickly through the cell. The node of Ranvier refers to the gaps in between the myelin sheaths on the Schwann cells, and they allow action potential to move quickly through the neuron and jump from Schwann cell to Schwann cell. The cell body is pretty self-explanatory. It's just like it's this part of the neuron that contains the nucleus. We have the dendrites that allow for cell communications. They are the receivers. And the axon terminal, which contains the neurotransmitters. Now we'll get into the process of cell communication a little bit later, but basically the neurotransmitters located in the axon terminal and the receivers located in the dendrite work together to transport information from neuron to neuron. A crucial part of the action potential is the sodium-potassium pump. The sodium-potassium pump enables the propagation of messages, also known as the action potential, to travel from one neuron to the next. When a stimulus occurs, action potentials travel across the axon towards the axon terminal. The way the sodium-potassium pump works is that it creates these action potentials all along the axon, so they're going in sort of like stages. And so it starts at the beginning, and then that one will complete the cycle, and then it will keep going until it reaches the axon terminals. And then from there, neurotransmitters are transported out of the cell, and cell communication can occur. An action potential occurs in phases. So the first phase is called the resting phase. Now the resting phase is when the inside of the cell is negative 70 millivolts. The sodium-potassium pump is on, and there is a steady flow of ions in and out of the cell. When the cell reaches its threshold of about negative 55 millivolts, the sodium channels open and there is a rapid influx of sodium into the cell. The sodium-potassium pump is on and the inside of the cell is getting more positive. This stage is called depolarization. In this next stage, the inside of the cell reaches a positive 40 millivolts, which is the threshold. And this causes the sodium channels to close and the potassium channels to open up, which causes the flow of potassium ions out of the cell. This makes the inside of the cell more negative. This phase is called repolarization. In this final phase, the charge of the inside of the cell goes below the negative 70 millivolts that we want our cell to be. So the sodium-potassium pump is crucial here. Active transport moves ions in and out of the cell to create this negative 70 millivolts again, and this stage is called hyperpolarization. So these action potentials are traveling down the axon extremely fast, creating these nerve impulses. The frequency of these action potentials all depend on the intensity of the stimulus that begins this action potential in the first place. The end of the neuron is where all the transport occurs. This is an image of the end of the neuron. So here on the top, we have the axon terminal. Now here we have what is called the presynaptic membrane. That's just the end of the axon terminal. And then on the bottom here, we have the dendrite of another neuron, and the membrane here is referred to as the postsynaptic membrane. This is because the space in between them is known as the synapse, and a lot of stuff happens here. Here we have vesicles full of neurotransmitters. And here we have calcium entering the end of the axon terminal through a voltage-gated channel. The entrance of the calcium ions through the gated channels stimulate vesicles to fuse with the presynaptic membrane, releasing the neurotransmitters into the synapse. These calcium channels are open because of the depolarization of the membrane caused by the traveling action potentials. There are receptors located on the postsynaptic membrane. The neurotransmitters bond with these receptors in the postsynaptic membrane and enter the postsynaptic part of the neuron. The neurotransmitters are either destroyed by enzymes or taken back into the presynaptic neuron. This taking back into the presynaptic neuron is called reuptake, and this occurs via active transport. We also have something called a reflex arc. So the reflex arc is involuntary, and it's a very quick movement in response to any stimulus. So this does not require a huge action potential because the body must react quickly, and going through the whole process would make the process too slow and we wouldn't be able to react in time. The next topic we're going to be talking about refers to the kidney. So the first question we're going to answer is, what is osmoregulation? Osmoregulation is the management of the body's water content and solute composition, and this all happens via the process of osmosis. Animals use this process to excrete metabolic waste products. We're going to focus on different types of nitrogenous waste. Now, the type of nitrogenous waste that is excreted depends completely on the organism and the water availability around them. For example, ammonia has a very high toxicity and is found in aquatic organisms because they're able to dilute that ammonia in water. Urea is found in humans because we are able to filter that out, and uric acid is found in flying organisms because they're also able to excrete that out properly based on the amount of water around them. The flying organisms have very low water content because they need to fly, so their waste product is in line with that. Osmoregulation in plants is similar. Plants like xerophytes, which are plants in dry habitats like cactus and succulents, have to modify their structures to adapt to their climate, and depending on their climate, they store water in different ways and osmoregulate in different ways. What we're going to focus on is osmoregulation in humans. Now, this occurs in the sodium-potassium pump, which helps to regulate the functioning of the kidneys, and it also occurs in the functional unit of the kidneys called the nephron. At the nephron, waste is filtered from the blood to produce urine. Here we have a picture of the human excretory system. So we have the kidney already labeled. These two tubes right here are called the ureters. We have the urinary bladder and the urethra where urine is excreted. These two veins right here, the blue one is called the renal artery, and the red one is called the renal vein. The kidney filters the blood. The renal artery brings blood to the kidney to be filtered. The renal vein carries blood away from the kidney, and the ureter carries the urine to the bladder. The bladder stores the urine, and the urine leaves the bladder through the urethra. In the kidney, we have these functional units called nephrons. They're located in this green area here. The nephron is specifically responsible for the filtration of blood into urine. The first step in this process is called ultrafiltration. This process occurs at the Bowman's capsule. The Bowman's capsule refers to this purple highlighted part right here. It's sort of like a cup holding on to the glomerulus, which is this cluster of red right in the center of that cup, the Bowman's capsule. The glomerulus is composed of a bed of capillaries. There are two arterioles attached to the glomerulus, the afferent arteriole and the efferent arteriole. The afferent arteriole transports small molecules such as sodium, potassium, salt, urea, and glucose, as well as large molecules, into the glomerulus. But the large molecule cannot fit through the glomerulus and do not enter the Bowman's capsule. Instead, they exit through the efferent arteriole. In the efferent arteriole, small molecules are not present. That is a main difference between the afferent and efferent arterioles. So a summation of ultrafiltration would just be removing large molecules from the bloodstream. And this occurs by pushing the blood through the glomerulus where the large molecules cannot fit. The group of substances filtered out by the glomerulus and the Bowman's capsule can be called a filtrate. The next step in this process is called selective reabsorption. The filtrate is transported to the proximal tubule where certain substances are reabsorbed into the bloodstream. Molecules like glucose, amino acids, and sodium are reabsorbed into the bloodstream. As we'll see in a little bit, not all the molecules that the blood wants are reabsorbed, so there's another reabsorption that occurs a little bit later in the process. The proximal tubule can also be labeled as the proximal convoluted tubule. The next step in this process is osmoregulation, which occurs in the loop of Henle. The ascending side of the loop of Henle pushes sodium out. The descending side of the loop of Henle pushes water out. The water is reabsorbed via osmosis. The cortex refers to the outer layer of the nephron, and the medulla refers to the inner layer of the nephron. The sodium that is released in the ascending side of the loop of Henle creates a hypertonic environment in the medulla. This creates a concentration gradient. The hypertonic environment that is created when sodium leaves the ascending side of the loop of Henle creates a hypertonic medulla. Now, this allows water to passively exit the collecting duct, which we will discuss in a little bit. The next step is another selective reabsorption, which occurs at the distal tubule or the distal convoluted tubule. Additional ions and water are secreted into the blood. So any molecules that the blood still needs that was not reabsorbed in the first selective reabsorption are now absorbed into the bloodstream. The final stage of this whole process in the nephron is another form of osmoregulation, and this occurs at the collecting duct. Antidiuretic hormone, known as ADH, regulates water. This ADH signals aquaporins in the collecting duct to release water into the blood. So when there's more ADH, more aquaporins open and let water reabsorb into the blood. If a person is very hydrated, then less ADH would be secreted because the bloodstream would not need as much water. If a person is very dehydrated, there would be a high concentration of ADH because we would want more aquaporins so that more water can be reabsorbed into the bloodstream if needed. So if someone is dehydrated, ADH would be released, and if someone is overhydrated, ADH would be inhibited. So in this final step, urine is created. So urea is excreted, H2O is excreted, and other salts are excreted. We have millions of nephrons in our kidney that create this large amount of urine that we produce. Without this process, we'd have a large amount of waste in our bodies. And to go over the process that occurs in the nephron one more time. First step is ultrafiltration. The afferent arteriole contains blood with small molecules and large molecules, and that goes through the glomerulus. The blood with large molecules exits through the afferent arteriole because it's not able to pass through the glomerulus. The filtrate that is created from the filtration in the glomerulus jumps into the Bowman's capsule. From the Bowman's capsule, the filtrate travels to the proximal tubule, where the small molecules are reabsorbed into the bloodstream, or whatever molecules are needed in the bloodstream are reabsorbed. Next, it goes through the loop of Henle. In the descending side, H2O is released, and in the ascending side, sodium is released into the medulla. This sodium release creates a hypertonic medulla, which allows H2O to exit the collecting duct passively. Then there's a second selective reabsorption, which reabsorbs any molecules that were not reabsorbed the first time. And finally, we have the osmoregulation in the collecting duct. So an antidiuretic hormone known as ADH is used to open up or close aquaporins, which regulates the amount of water in the urine and in the bloodstream. So when there's a high amount of ADH, that means that the bloodstream needs more water, so more water is released into the blood because the aquaporins are open. When a person is overhydrated and does not need water, the ADH is inhibited, so less water is diffused into the bloodstream.